Hi, I'm Dr. Jay Dunn, and I'm super excited to be with you guys here at the ICAK. This is my first time lecturing for the ICAK, and I'm just thrilled. So thank you, Dr. Belli, for inviting me, and I'm thrilled to be among such amazing practitioners and very smart people. You guys are the best of the best. So thank you very much. This is a re-recording of my lectures. Something went wrong with the recording of the original lecture, so I'm going to try to remember what I said. So my aim today is to take you through an understanding of how you can work with genetics with your patients. We use methylation cycle assessment as a term, but it's really more than just the methylation cycle. We're looking at a bunch of different biochemical pathways that intersect each other, and all of them, kind of are impacted by the methylation cycle and also impact the methylation cycle. So you'll see how they all interrelate. So my goal is to kind of take you deep into understanding biochemistry and looking at health in a very different way. When I started doing this work, it just changed everything that I knew, everything that I thought I knew about the body. It changed. So it was a big paradigm shift. So I'm hoping to give you that sense of looking at the body in a very different way in just an hour. I have a lot to say, so I'm going to be flying through some of the biochemistry and assuming a lot of knowledge here, but I want you to get sort of the sense of where I'm going with this. The whole time, with this understanding these pathways, I'm using functional muscle testing. I do a technique called Wholistic Kinesiology, which is one I developed after learning all the different types of kinesiology. I love kinesiology. It's my passion. It's my life. I just can't imagine living life without understanding how to muscle test and find out what's going on in your environment. Just to give you a little of my background, I'm a chiropractic physician, but I was doing the kinesiology before I became a chiropractor. So I've been a kinesiologist for over 30 years, since 1988. And again, I'm very passionate about it, and I love it. My lineage of learning kinesiology came down through Beardall. My teacher studied with Beardall, and I studied with my teacher. Again, before I even became a physician, I was doing some kinesiology and having amazing results with all kinds of conditions, and it got me excited. It got me excited about understanding the body and understanding it to the nth degree, as much as I can learn about the body. And I know you guys are this way too. Anything you can learn about the human body is going to make you a better practitioner. So I was always looking for answers, and I had some cases where I couldn't get anywhere with people, and I have a lot of tools in my bag. I'm a functional medicine practitioner. I'm a chiropractor. Went to Palmer. I do a lot of emotional work. I do EFT and NET and emo-free and like every technique I could get my hands on, NLP. And I still had people that didn't respond to anything I did with all of these tools in my bag, and so those are the ones, as you probably know, those are the ones we take home and we think about, and we're like, what am I missing? What's going on here? And I knew there was a piece missing because for myself, I also struggled with my own health. I had health issues, chronic fatigue syndrome of over 40 years, and depression, lifelong depression that I was always struggling against. And so I knew there was another piece of the puzzle that we hadn't gotten to yet, and so I kept looking. The other piece that kind of gave me pause was my father, who died of lung cancer at 55, which was not a big mystery. He did everything he could to get lung cancer, I think. He was on a mission. But the mystery was that my stepfather, my dad's best friend, ended up marrying my mother after my stepfather died, and he did the same things: smoke, drank, stressed out, no exercise, no health habits whatsoever, and lived another 30 years doing those same things that, caused my father to die of lung cancer. And so I thought that, the only thing that we can contribute that to is genetics. And so for the longest time, we just kind of parked genetics over to the side and said, we don't know how to work with that. We just get to blame it when things don't work. But the more I looked into this, and the more I heard this word methylation and started looking into genetic variants and their effect on our health, the more I was convinced that this was indeed the missing piece. So I want to give you a sense of that journey and the information that I found when I started looking. All right. So, again, we kind of commend ourselves for not looking at symptoms. We try not to treat symptoms, which are on the top half of this chart, but we've been looking at mental and emotional stress, nutritional deficiencies, physical trauma, toxic overload. We've been looking at the soil or the constitution of the body. And so that's served us well, but still, I still think we've missed that one little piece that can have an effect on all of those things, that can increase mental and emotional stress and increase nutritional deficiencies and cause you to be more susceptible to physical trauma or toxic overload. And that is the genetics. So we need to remember that there's that one other piece in this puzzle. That's not to say that genetics sits alone by itself as a cause. It is definitely the combination of genetics, environmental inputs, and lifestyle behaviors that are underlying factors that contribute to chronic symptoms and illness. So we have to remember it's that interplay of genes and environment. That's what we call epigenetics. This is all new. The Human Genome Project determined the sequence of chemical base pairs which make up the human DNA and of identifying and mapping all of the genes of the human genome from a physical and functional standpoint. This is the last chromosome was finished in 2006. So this is a very new science, and every day, there are new studies showing, what genes are associated with what particular a risk of certain imbalances in the body. Well, that's cool. I mean, it's going to be an ongoing process, but there are certain fundamental genes that we can look at that have a very real effect on things that we're looking at as, functional medicine doctors, as applied kinesiologists. We look at things like toxicity issues. How well does a person detox? And that can be very much genetically determined. You can have genetic variants in phase 1 and phase 2 detoxification pathways of the liver. Or you can have genetic variants in the Krebs cycle, in their ability to turn food into energy. Or you can have genetic variants that don't allow you to make neurotransmitters correctly, serotonin and dopamine being the prime ones, which can cause lifelong depression. Or you could have genetic variants in the nitric oxide pathway, which can lead to high blood pressure. So these are areas, what we call the hub genes, that we want to pay particular attention to, and they are enzymes. So I want to talk to you more about that. Let me just show you how this works. We take the genes out of the chromosomes. We have 23 pairs, as you remember. And if we unwind them, we're looking at the base pairs, if you remember those in cell physiology, the A, T, C, and G. And those make up our code, our blueprint for understanding what that DNA is coding for, or what that gene is trying to tell our body to make. That's the blueprint or the recipe. And then from that, the RNA and then, takes a copy, and then we produce our proteins from that genetic code. So if that genetic code has a change in it or is a variation from normal, then that protein or that enzyme is not going to function the same way as if we had the correct recipe for that enzyme or protein. And that's not to say it's a mistake. It's just a variation. It's what makes you unique and different. For instance, you could have a genetic variant in your ability to make the protein that breaks down serotonin, which makes you more grouchy than the normal person. It doesn't mean that's wrong. It just means you're more aggressive or more of a warrior. That's what we call the warrior gene, the MAO. It makes you a different kind of person than somebody who can break down their serotonin efficiently. So it's the differences in personality and in how we think and in how we act and how our energy works and how well we detox, or how well we can how resilient we are to the environment. So how many genes do humans have? Genes vary in size from a few hundred DNA bases to more than 2 million DNA bases. So they can be very small or very huge. The Human Genome Project has estimated that humans have between 20,000 and 25,000 genes. So if you do the math there, if we had 20,000 genes at around 1 million DNA bases each, actually the number ends up being around 6 billion base pairs that we have, and that's a lot. So it's a lot to understand, and we don't need to know all about those 20,000 or 25,000 genes. We're looking at some very critical genes and how we absorb nutrients or how well our body works. So the complete genome scan would be 6 billion A's, C's, G's, and T's. The protein coding, including enzyme sequences, account for only a small fraction of the genome, and that's what we're interested in, approximately 1.5%, around 300 genes. The rest are associated with non-coding RNA molecules and sequences for which as yet no function has been determined. So again, ongoing research here to figure this out. Go. So the exome, that's what we're looking at. Proteins make up our physical structure, catalyze the chemical reactions that keep us alive, and regulate the expression of other genes. The vast majority of heritable diseases that we currently understand involve tiny differences in one of the exons that collectively make up the exome. So that's what we're going to be looking at. Enzymes, let's talk about those for a minute. Enzymes are proteins that accelerate or catalyze chemical reactions. Now, we're not just talking about digestive enzymes, we're talking about other enzymes that increase the production of taking a substrate and making a product out of it all throughout the body. Everything that happens in the body requires enzymes. So the molecules at the beginning of a process are called substrates, and the enzyme converts these into different molecules called products. Almost all metabolic processes in the cell need enzymes in order to occur at rates fast enough to sustain life. So if that enzyme is so inhibited that it can't sustain life, then you don't survive. So obviously you have enzymes that are working, but it's all about speed. So if you have enzymes that aren't working up to 100% of their possible function, then that pathway or whatever that enzyme is supposed to do is going to be slower than normal. So for instance, if you're looking at some of the enzymes that break down our food and make ATP or energy, and you have inherited a genetic variant in those enzymes, then that whole process will be slower than normal, and you'll tend to gain weight and have lower energy. Makes sense, right? So this is from Bruce Ames, Department of Molecular and Cellular Biology, University of California, Berkeley. He's one of my heroes. He's doing this work. He's talking about supplements, and he says about 50 human genetic diseases due to defective enzymes can be remedied or ameliorated by the administration of high doses of the vitamin component of the corresponding coenzyme, which at least partially restores enzymatic activity. Our analysis of metabolic disease that affects cofactor binding, particularly as a result of polymorphic mutations, may present a novel rationale for high-dose vitamin therapy, perhaps hundreds of times the normal dietary reference intake in some cases. This area should interest the entire health community because of the considerable percentage of the population affected by polymorphisms, many of which have outlived their genetic usefulness. So every one of us has some of these genetic polymorphisms. So the interesting thing is that we can work with them. We can understand those polymorphisms or enzymes that aren't working correctly and give that person higher levels of cofactors to make up for this missing enzyme or a need for higher levels of certain nutrients to make up for this genetic variant. And then we can restore normal function to an enzyme. So that's pretty exciting. This is from Roger Williams, another pioneer in the field of biochemical nutrition. He also recognized that higher dose of vitamins may be necessary to accommodate for what he calls biochemical individuality. Individuality in nutritional needs is the basis for the genetotrophic approach and for the belief that nutrition applied with due concern for individual genetic variations, which may be large, offers the solution to many baffling health problems. So again, understanding that each individual may need higher than normal doses of a particular nutrient to make up for their particular genetic pathways. So this is DNA damage. You can have a single strand break, a mismatch, a damaged base, double strand breaks, cross links, etc. This happens all the time at the cellular level, and what happens is if your methylation is not fully operational, if you have genetic variants there, then you won't repair this DNA damage, and that can cause a long-term issues. So I'm going to get out of this for just a moment, and I want to take you to these pathways here. This is what we're going to be looking at. Let me play this one here. We're going to be looking at these individual pathways in the body. So on the left-hand side, you'll probably remember the Krebs cycle, or mitochondrial function is over there. It's how we turn food into energy through the process of the Krebs cycle and the electron transport chain. So some of the main genetic variants that we're looking at there are the ACAT, the MMAB, and the NDUFS, I call it, the NDUFS. You can have there are more than these in there, but I just wanted to give you the highlights. So if you have inherited a genetic variant in any of those enzymes, it will slow down this process. And it makes sense that this person would have low energy and would tend to gain weight. They also would have high cholesterol and a risk of cardiovascular disease, along with neurological diseases. Mitochondria is super important in understanding mitochondrial function or energy production in the body. So what happens is you get one gene from mom, one gene from dad. So if both of those genes are the normal code for a particular enzyme, then that enzyme is going to work at close to 100%. If you have one gene that's a variation and one gene that's normal, that's what we call a heterozygous variant, and that's a SNP, or a single nucleotide polymorphism. If you have both genes that are a variation from normal, then that enzyme is what we call heterozygous or homozygous, and it can be reduced by up to 70% of its normal function. So that means it's only working at about 30% of normal activity. That enzyme is really sluggish, and so you can see where that would have an impact on how well the body's going to turn energy into food, or food into energy. So we look at those things, what you inherited, and they're not mistakes. Again, they're just variations. So if your ancestors were alive during times of famine, and they inherited a gene that allowed them to slow down their metabolism, then they would survive during times of famine. However, they passed that gene on because they did survive, and their subsequent generations are going to have a hard time losing weight. They're going to hold on to weight very easily. Very frustrating. The second pathway, we're looking at cardiovascular circulation and blood flow. This is the nitric oxide pathway and the inflammatory pathway. So we look at nitric oxide synthase, the NOS gene, and that one is how well we make nitric oxide in the body. And if you can't make nitric oxide efficiently, then you're going to have things like high blood pressure, because it's going to squeeze down the blood vessels. Nitric oxide should dilate the blood vessels and allow blood flow to happen. If we can't make nitric oxide, it's going to squeeze those blood vessels down. Makes sense that would lead to cardiovascular circulation issues and high blood pressure. So a high risk of cardiovascular disease when you have that genetic variant. It's also how we make peroxynitrite and superoxide, and these are highly inflammatory molecules. If we have a genetic variant in that SOD gene down here at the bottom, then you can't break down these highly toxic and inflammatory molecules, the peroxynitrites and superoxides. These are not mistakes. The body makes those in order to fight off infections. That's part of our innate immune system. It's how we kill viruses, bacteria, yeast, fungus, parasites. And so without those, if you can't make those nutrients in the first place, then you tend to get infections easily. So we have to look at, the interplay of these genes here. The next pathway, this one right here in blue or purple, I guess it is, light blue. These are the brain hormone neurotransmitters pathways. So we're looking at tyrosine being made into dopamine, tryptophan being made into serotonin. So if you have genetic variants along here or here, then you're going to have low levels of serotonin or dopamine and tend to have depression. Depression, we know, has a very strong genetic component to it. And the more we understand this, the more we can see why that actually happens. And then down here is how well do we break serotonin and dopamine down, and those rely on these two enzymes down here, MAO and COMT. If you have genetic variants there, then you can't break dopamine, norepinephrine, or adrenaline down correctly, and you can have high levels of serotonin or dopamine, and that can cause ADD and anxiety, aggression, irritability, inability to just calm down. This is your it's stress. These are your stress monsters. This is sympathetic dominance here. So there's a genetic reason for sympathetic dominance. And then down here, we're looking at how well does glutamate convert into GABA, and that depends on that enzyme right there, the GAD enzyme. Genetic variants there cause the person to hold on to glutamate much too readily, make them very sensitive to monosodium glutamate, which can lead to neurological dysfunction. Glutamate is a neurotoxin or an excitotoxin, and low levels of GABA. So this person is also anxious, and prone to neurological diseases, and also seizures. Seizures are there as well. So over here in this pinkish magenta area, we're looking at the vitamin D receptor, and this is a big deal. This vitamin D receptor, and that, when I first started looking at genetics, this was one I focused on because it was one I had. And when I started Googling it and looking at PubMed and all the studies that had been done on the vitamin D receptor, I was shocked. But I was also incredibly motivated to understand it better, because for several reasons. One is, this is the receptor for vitamin D. It's not how well you absorb it into your body, it's how well that receptor can take in vitamin D. And so as a kinesiologist, this was always confusing to me, because I'd see patients that had low vitamin D levels on their blood work, And then I would go and try to give them vitamin D. I'd muscle test them, and it wouldn't test strong. The body would say, no, don't give me vitamin D. And it always like, what's going on there? I'm scratching my head going, what? Wait, their vitamin D levels are low, but their body's saying, no, don't give me vitamin D. I know AKs work, so it's not a mistake. It's telling me something. So I went back to the drawing board and started looking at that vitamin D receptor, and it's on every cell. It's on the nucleus of every cell in the body that has a nucleus. And so it's, it's ubiquitous, but it needs cofactors. So what we think happens is when you take vitamin K and you take vitamin A with the vitamin D, it changes the conformation of that vitamin D receptor, and now vitamin D can get in. So it seemed to upregulate your ability to get vitamin D into the system. So it's not just, do we get it in the diet? Do we get a liposomal form? Do we get it from sunshine? It doesn't matter if those vitamin D receptors aren't working correctly. It's not going to get into the cell and have the effect that we want. It has a very profound effect on the immune system. Huge. It's how we mature T cells in the body. And without mature T cells, you can't fight infections thoroughly. So for these people, you're going to be fighting infections all the time. It's like whack-a-mole. You got virus now, you got parasites, you got bacteria. This person has always got some kind of an infection. And so that means that the immune system isn't maturing the T cells correctly to be able to fight off infections. So those are two areas where you would want to look at really upregulating the immune system is the nitric oxide, the inducible nitric oxide synthase, and the vitamin D receptor. Very critical. Also, the other thing that vitamin D does, once you get it in the system there, is it turns on your ability to turn tyrosine into dopamine, tryptophan into serotonin. So it affects the pathway next to it, which affects the pathway next to it, which affects the pathway next to it. These all affect each other. They're not sitting there in isolation. So when you turn on the vitamin D receptor, the body makes more dopamine and serotonin. Here's the problem, though. When I talk to a lot of people about, I'm just going to go in and work on that vitamin D receptor. Don't do it. Because now you've made much more dopamine and serotonin, but do you know that person can get rid of it? Do you know that MAO or that COMT gene can actually break down the extra dopamine or serotonin that you're making? Otherwise, you're going to send them into aggression and irritability and ADD and anxiety, and they're not going to like you very much. So you have to look at how genes interact, and that's really critical. I want to talk more about that in a minute. The next pathway over here, the orange one, this is liver detox area. So if you have genetic variants here in the CBS, SUOX, SALT, the cytochrome P450s are here, UGT, which is phase 2. So you can have genetic variants all through the detoxification pathways, which makes these people much more susceptible to toxicity issues. Heavy metals, chemicals, pesticides, glyphosates, etc. Can't be broken down efficiently. And so you're always having to do a detox program on them instead of looking at how do we work with their genes to get them to work better and upregulate their detoxification. That's, I think, a better question. And then the pathway above it here, we're looking at the methylation cycle in yellow. And genetic variants here can impair our ability to turn on methylation. So let me talk to you about methylation for just a minute. This pathway on the left-hand side, this circle here, is how we turn folic acid into 5-methyl tetrahydrofolate, the active form of folic acid. So this MTHFR is the most well-studied and thoroughly researched genetic variant, and the mother flipper, we call him. And genetic variants there will keep you from being able to activate folic acid, that's a real problem. And the one next to it here, this is the homocysteine pathway or the B12 pathway. So they actually bump up against each other and they turn much like cogs turn, and they have to be both in sync in order to make methyl groups. And that's an important thing. Methyl groups, when we make them, they're going to lower homocysteine levels. You can see homocysteine right here. Homocysteine will then be made into methionine and then into SAMe, and then we turn on methylation. And SAMe is our main methyl donor in the body. I'll talk about him for a little bit here. But one of the main things that we're going to do with that methylation is turn on RNA and DNA repair. And as I was mentioning a minute ago, we get DNA damage at all times of the day and night. And if you're not methylating correctly, if you have genetic variants there and you can't repair that DNA, that's a problem. So it does so much more than that, but that's one area that we want to look at. But for instance, this enzyme down here, the catechol-O methyltransferase, the COMT. It's a methyltransferase. It requires a methyl group to transfer adrenaline, conjugate it, and break it down, essentially, and get it out into the urine. So without methylation, this enzyme is not going to work correctly. And that's true of a lot of enzymes throughout these pathways. They require methyl groups to really work correctly. So you can see this pathway affects all these other pathways by donating methyl groups. And over here on the left, in that mitochondrial area, that guy is donating ATP to all these enzymes. And so each of these enzymes require ATP, a cofactor, and a methyl group to really kind of upregulate the ability of that enzyme to work closer to normal function, even though we've inherited a genetic variant there that can inhibit that. So that's kind of a little bit of an overview of what we're looking at there. I'm going to pick out some other slides in here just to give you an idea. So what is methylation? When we look at methylation, it's basically a carbon and 3 hydrogen molecules. So you can see it right here. And it attaches to a base pair. These happen to be an RNA bases here. But it changes the conformation. So where that H should be, it actually now a methyl group is attaching there, which has an interesting effect on the body. So we look at genes. So here's the chromosome, again, being unwound, and here's a methyl group being attached to the DNA. And what happens is it causes that DNA to wind around something called a histone tail. That DNA is now hidden, that gene is turned off and cannot express whatever it was going to express, whether that's a cancer, or heart disease, or some kind of a birth defect. That gene is now not going to be able to express. And over here, this DNA is accessible, the gene is turned on because of an acetyl group. So we have this combination of methyl groups and acetyl groups turning genes on and off all the time so that they can express correctly. So underneath it says small molecules like methyl or acetyl groups can switch genes on or off by binding to the DNA or to the histone proteins it winds around. Methyl groups act like a roadblock, stopping enzymes from reading the DNA, while acetylation can loosen the DNA coil, exposing hidden genes so they can become active. So it's just a profound thing that happens in the body, and without methyl groups, we can't, things are not going to function normally. And here's one way you can look at the methyl groups as well. That's super important. So when we're trying to turn norepinephrine into epinephrine, so epinephrine comes from norepinephrine, and that comes from tyrosine. So we break down into dopamine, norepinephrine, and epinephrine. This pathway requires S-adenylmethionine, the SAMe that we talked about, which is a methyl donor. So it drops a methyl group off and becomes S-adenosyl homocysteine. And it allows this phenylethanolamine N-methyltransferase to turn norepinephrine into epinephrine. This also requires B5 to function correctly. So that's its cofactor. Also requires ATP. So we have to look at, are we making ATP correctly? Do we have enough B5, pantothenic acid? And do we have enough methyl groups to turn norepinephrine into epinephrine? If you can't do that, here's your patient that has a low adrenal glands. This person, when they stand up too quickly, they're going to get dizzy, and orthostatic hypotension happens there, right? If they can't make epinephrine or adrenaline, how many of our patients do we look at that are adrenal patients? Almost everybody I saw in my practice needed adrenal support. And so this is a big deal. If you have a genetic variant here and you're not looking at that piece of it, then you're giving them adrenal support instead of getting their body to make it. And that's where I think that's the paradigm shift that I want you to get, is that, our body, our adrenals are just sitting there going, man, if I had a little methyl donor, if I had a little P5 or a pantothenic acid and a little more ATP, man, I could make some adrenal. I could do it all myself. And those things are, naturally occurring in the body. We're so used to thinking that, we have to patch the body back together instead of giving it what it needs to really do its own job. And that was the paradigm shift. And that's where, I love the concept because, in chiropractic, our philosophy is that power that made the body can heal the body. It just needs the right nutrients to do that. And for this person, they need some SAMe. They need some pantothenic acid. and make sure that their mitochondria is functioning correctly, and then they can make adrenaline instead of giving it to them. And it just made a lot of sense to me. So I'm gonna get out of here for a second because I have such limited time. I want to go to the key areas. Oh, here's another area that I think is super important. The liver detoxification pathways, and we're looking at phase 1 and phase 2. This is cytochrome P450, and this is conjugation pathways over here. But if you take a look at the nutrients that contribute to phase 1 cytochrome P450 enzymes working correctly, they are riboflavin, niacin, pyridoxine, folic acid, B12, glutathione, branched-chain amino acids, flavonoids, and phospholipids. And if you have genetic variants in your ability in methylation and your ability to activate folic acid and B12 right here, it's going to impair your detoxification. If you have cytochrome P450 enzymes that you've inherited and you have a genetic variant there, it's going to impair your phase 1 pathway. Over here in phase 2, we're looking at sulfation, glucuronidation, glutathione conjugation, acetylation, amino acid conjugation. Again, you can have genetic variants in phase 2, which are going to slow down your ability to detox and go through these pathways and conjugation pathways. So you can see where a genetic variant could have a very real effect on your ability to detox, for sure. Oops, let's do that again. There we go. Okay, so it's kind of a big deal. So again, this is a SNP, what we call a single nucleotide polymorphism. How we turn a T, like this is our normal code for a particular gene, and you inherited instead a C where the T should be. That's going to impair that enzyme's function. So that's what we refer to as a single nucleotide polymorphism. Again, a heterozygous variant will reduce that activity of that enzyme by about 30%, homozygous by about 70%. And that's a big hit. That's a tough one to get around. Okay, so when it comes to genetic testing, in the past, we've been looking at Ancestry. We've been using Ancestry DNA test kit. It's only $79. It's simple, inexpensive, and you just spit in a tube and send it back. Well, we found that the data that we were getting is not very complete. We're missing quite a few genes that we're interested in looking at. And same with 23andMe. Those were the two that we were kind of using. So we decided, our team here decided to go ahead and get our own DNA testing going. And so this is what we're going to be using, is the My Happy Genes genetic test. It's completely private. That's the other issue with a lot of genetic testing, is that there's a privacy issue. 23andMe and Ancestry sell your data, and they sell it mostly to pharmaceutical companies. And we're looking for that increased information. It is going to be slightly more expensive. It looks like we're coming in at retail at $250, but we have almost twice as many genes that we're looking at in this test, and it's easier to do. It's a little swab. So it's here's our little test kit here. And it's just a swab, and you just, you go in, instead of having to spit in a tube, you just go in and swab the cheek, and you're sending that back in. That should be available just about any day on myhappygenes.com, the website. So we're super excited about that. The raw data that you get back looks like this. So with the Genome Project, these numbers over here, this RSID number, take a look at that. These are the identification numbers for every gene that they test for, that this company tests for. This was Ancestry one here. So they only test for a small amount of your data. It isn't your full genome. That would be a huge file. They're looking at whatever genes that they consider important to look at. But in this column right here, these are the ID numbers of those particular genes. And I don't know what those mean. I can't really read that. I didn't memorize the genetic variants. But these are all in chromosome number 1, and there's its position on that particular chromosome. And the allele 1 and allele 2 are here, what you inherited. Again, one from mom, one from dad. You got 2 Gs here. I don't know if that's the correct form of that particular enzyme or not. So we need a program that can interpret that information and give us something that we can use clinically. And so again, created another a program called My Happy Genes, and it analyzes that particular genetic data. So if we look up here, this particular gene here, the ACAT1, is involved in it's in the mitochondrial pathway. It's how you turn energy into food. And so we have several alleles. You can see there are 5 of them here, 5 different forms of that gene that we have. And so each of them have kind of different effects on the body. Some of them are in the energy production pathways. Others can cause mood swings or depression, brain function imbalances, premenstrual mood imbalances as well. And over here, the risk allele, the wrong code for that particular gene is A, and this person inherited a GG, which means it comes out green. You got the right results. Green is good. If you've got the risk allele is C, and you've got one gene from mom or dad, we don't know which one is which, and that's what we call a heterozygous variant, it'll come up yellow. But if you have both genes are the wrong code, like this one, it'll come up red. And again, that one's reduced by about 70%, so it can lead to these risks of mood swings or depression over here. And when you look at this report, if you click on this, it'll open up a balloon. It'll tell you exactly what that gene does and where I got this information. I have links to studies from PubMed that show that that gene is involved in mood swings or depression, or where I got that information from. So it's a very comprehensive amount of information. We have probably 500 genes that we have on this report at this point. I keep adding them almost every day. I'll add a new one. So you remember the Punnett square of inheritance. It's kind of a crapshoot of what you'll get. So you don't know. You could be very different than your spousal and than your siblings as well. So we use kinesiology techniques to really kind of identify these genetic variants. And it's really cool because you can use the kinesiology to spot the genetic variants before you ever get the gene test back. And the accuracy is amazing. We found it to be incredibly accurate. So I'm going to show you just a quick demo of how we use the kinesiology techniques to find out if there is a genetic variant or not. We're going to use the vitamin D receptor, the one I told you about, and show you how we do that. So these are some kinesiology techniques that I find very helpful, and in the demo we'll show you how to use those as well. All right, so they're in your notes. There's some information about safety, beneficial point, preference. These are very cool points, and I encourage you to take a look at them and use them because I find them to be incredibly useful in doing this work. So when we're testing for a SNP, or a single nucleotide polymorphism, we're checking the substrate. In other words, like the noradrenaline, and then the product would be adrenaline, and the enzyme there is the PNMT, phenylethylalamine N-methyltransferase. So we're looking for the presence of a possible SNP or enzyme defect that will inhibit the ability of that enzyme to complete the reaction of substrate to product. This causes the substrate to be too high, and it's going to test adverse, and the product will be too low. It's going to test beneficial. So noradrenaline would be too high, adrenaline would be too low. And then you can go, okay, I think this is a genetic variant here. And then to confirm it, you would go and look at their genetic results. All right, so let me just show you that. I'm going to show you a quick demo on that right now. Okay, so I'm just going to do a quick demo because the technique is actually a lot longer than we have time for. But I just wanted to give you a sense of how the flow would go with using kinesiology to kind of find where genetic variants are. These are our test kits, 1 through 10. But for the basic protocol where we're going to start, I always start with the basic test kit. So these are energetically frequenced vials. The frequencies are encoded into the vials so that they're the energetic equivalents of the substrates that we're looking at. Just to remind you, so this is the same pathway that I showed you earlier on the interactive chart. And it's a way to kind of keep track of where you found sort of a slowdown or a genetic variant in those pathways. So the testers are in the order of clearing. They're going to go through, start at number 1. And I showed you those numbers on the chart there. Number 1 starts down here in the electron transport chain, looking at how well do we make energy out of our food. And then it goes, through the pathways in a particular order. It's really important that you do it in that order. We have found through trial and error that if you go off of that order, you're actually can cause a lot of problems in a patient, because you're upregulating a different pathway, and it's going to need support before you can upregulate it. So you have to do it in a particular order. And so this kit and the whole system, the worksheet that we're going to use is also in the order of clearing, and I showed you that a minute ago. You can follow along the initial appointment protocol to kind of figure out how to do this. But basically, once you have the patient cleared, of course you test for switching and, whatever it is that you do to get your patient ready. Once you do that, what we're going to be doing is I'm going to have her contact these vials and go down the rows here. And when she touches one and it goes weak, then we know we kind of have a backup in the system. So let me just show you. So I'm going to have her touch that first vial, which is the ADP vial. And she's nice and strong. Cytochrome C is number 2, succinate, FADH, NADH, etc. So she would go down the lists until we find, let's go over here to infections, because I want to show you what would show up if you did have the VDR variant. So she would go weak on infections and T cells, potentially tryptophan and tyrosine. The other indication that she could have a genetic variant there is just to take vitamin D. We have the nutrient kit here. You can take vitamin D out of there, put it on the body, and test her, and she goes weak. So we know that she's not absorbing vitamin D. So we want to get her to absorb that. You can also just do it with a regular vitamin D supplement. So I could put vitamin D on her, and if that makes her weak, we have a good suspicion that she has a vitamin D receptor variant, because her body's saying, don't give me any vitamin D. I can't do anything with it. So what we have found is you have to use the cofactors. So we would use vitamin A and vitamin K together. I like a Complete K from NutriWest, and I like the pure brands. It doesn't matter what brands you use, as long as you get like the whole set together, vitamin K, A, and D, and get it to go strong. So lots of different brands could do that. So we're adding those cofactors in there, and then that makes the vitamin D strong. So we know that she needs these 2 cofactors to get that in. Again, a strong indication that she might have a vitamin D receptor variant. I'm just going to stick those next to her real quick. And now I'm going to have her go back and touch that vial. Let me see your finger. We're going to touch right here on infections, nice and strong. T cells, nice and strong. Tryptophan and tyrosine are now strong. So we know that we've actually fired up her immune system. We're getting her body to fight infections rather than trying to go in and take an antiviral or take an antibacterial, find out what infections are there. Often, if I find one infection, I'm going to find a bunch of different infections because the door is open. Her immune system isn't really fighting anything off. So we would put those remedies in there, and then we'd go down the list the rest of the way. Again, I'm not trying to teach you the whole protocol right now. I just wanted you to get at the concept of how you spot a genetic variant. And in this case, we spotted that vitamin D receptor through this system. This is a level 1 material here. The first kit is the organ sweep kit. The second one is the basic test kit, and it takes you through that kit takes you through the entire pathway here. And then we get into the advanced classes. We get into advanced methylation, the hormones, allergies, cardiovascular kit. This is a super challenge kit, the emotional test kit, and then the last one are the supplements. So these are different brands of supplements that can help balance out these pathways whenever you find an imbalance there. So, all right, thank you for your attention. Welcome back. So a couple other little points here. One is one does not dabble in methylation. Here's the problem, that when you start upregulating a particular enzyme, it's going to cause a change in function of another enzyme. They all kind of interplay with each other, and you have to understand that. Trying to just work on one gene is very short-sighted. I see a lot of people trying to turn methylation on by giving somebody methylfolate, and I just cringe because there are so many things that are going to happen now. You've just turned on a whole bunch of knobs in the body, and you don't know the ramifications of that. One of the main things that happens there is that you turn on detoxification pathways too hard. That person is going to feel terrible. We did it. We really did this in the beginning and made a lot of people sick, including myself. So that's why we discovered that there's a particular order that you have to go in to get the body balanced all the way. And so let me show you how that works here. So this is my interactive chart. I created this chart so I could really remember all these details in the body. So it's a complicated bunch of pathways for sure, but every piece of it is defined. So if we go over here and we look at this vitamin D receptor, you click on that and it'll pop open a balloon. This chart is actually available for sale if you're interested in studying these pathways. It's an invaluable resource. So the vitamin D receptor enzyme is expressed in most tissues of the body and regulates intestinal transport of calcium, iron, and other minerals. Vitamin D increases the activity of tyrosine hydroxylase and therefore increases levels of dopamine. It's involved in the absorption of vitamin D, bone formation, immune system function, neurotransmitter function, etc. Low-functioning vitamin D receptor can lead to T-cell dysfunction. This in turn can lead to autoimmune conditions. Any improvement in vitamin D status will significantly affect expression of genes that have a wide variety of biological functions of more than 160 pathways linked to cancer, autoimmune, and cardiovascular disease which have been associated with vitamin D deficiency. So when I went on PubMed, I found studies associating genetic variants in the vitamin D receptor with all of these things. Rickets, hyperparathyroidism, cancers of all types, brain tumors, dental issues, chronic infections, degenerative disc disease, hypercalcuria, spinal stenosis, spondylitis, syringomyelia, osteoporosis, osteopenia, etc., etc. You can see the list is pretty freaking long. It's a lot of things. So blood work, recommended vitamin D3 levels in serum. Actually, vitamin D, active forms of vitamin D3 would be even better. ANA test could possibly tell you there's an autoimmune thing going on. But in kinesiology, our challenges would be T cells. And in the kit that I showed you during the demo, we have T cells, we have vitamin D in there, we have infections, insulin, L-DOPA, lactose, phenylalanine, tryptophan, and tyrosine. Those will all indicate that you have a low-functioning vitamin D receptor. And so diet and lifestyle recommendations, of course, getting sunlight, using Dr. J's healthy rays so you don't block those beneficial rays. That's a whole nother story. I created a non-blocking sunscreen. And we balance the VDR with vitamin K2, K27, K24, K27, vitamin A, vitamin D3, boron, manganese, calcium, manganese blends, and trace minerals. But also looking at how well do you absorb your fat-soluble vitamins. So you'd want to look at gallbladder function there as well. And there are some NutriWest remedies on there as well. And those are going to be changing soon. So every piece of this pathway is defined. Every word in here, if you pop it open, it'll give you a definition of what that means. PDH. So you can learn a lot in here, and there are lots of charts. This one is one particular chart. I got to say a word about this because it's so incredibly important. We're down in the electron transport chain, and we're looking at where energy is turned, or food is turned into energy way down deep into the electron transport chain, that last place there. But lots of things can interfere. We have complex 1, 2, 3, 4, and 5. This is oxidative phosphorylation. This is where we turn ADP into ATP, the final step there. But lots of things can interfere in this pathway, including electromagnetic fields, radiation, heavy metals, antibiotics, fluoride, chlorine, metformin, insecticides, antibiotics, medications, mercury. When I looked up the medications, I was stunned at how many different drugs interfere and cause mitochondrial damage. So that was kind of an eye-opening thing. So in my class, I study, I talk to you a lot about understanding these particular genetic variants and how they work. So there's a lot to learn here, and it looks pretty complicated, but here's the take home. I gave you a list, a couple of lists. One is the methylation worksheet. And so in that test kit that I showed you, you go down the challenges. They're all in a particular order, from challenge number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So if you follow the order, you'll do this in the right order. And if you just follow this worksheet, it's a very easy to use system. You just go from step 1 to step 2 to step 3. If this ADP vial tests weak, that's an issue in the electron transport chain. And these are the possible remedies that could balance out that electron transport chain. Same with the FADH and ADH. If that's weak, that tells us that you're not making energy out of your food. When you get to carnitine, this is indicating an SLC22A4 gene. Malonic acid is indicating an MMAB gene. And so these are the remedies that you would use. So I showed you just briefly the test kits that we use, but we also have the remedies in the kit too. So it's super easy to go, okay, this is weak. Look at these remedies and see what strengthens it, and then move to the next one. So let's go back real quick to that interactive program. And you can see the numbers here. We've done this in a particular order because we found we have a whole lot less side effects if you do things in a very specific order when you're balancing. So we start at number 1, number 2, number 3. Turns out they're in numerical order. And so if you balance that Krebs cycle ATP production first, and then you come over here and start balancing this area that has to do with how we make hydrogen peroxide and break it down, superoxide, peroxynitrite, the inflammatory free radical damage area. That comes next. We balance that next. 4 and 5 are there. And then we go up and take a look at nitric oxide pathway. Make sure that's balanced. And then we come down here to the glutamate conversion to GABA. That's number 7, number 8, etc. So if you stay in that order of clearing, which the worksheet guides you in, as does the test kit, bam, you got this. And so it's an easy to use system. Understanding the biochemistry behind it takes a little time, and it's a bit complicated, but it's really fun. When you start to work with this using kinesiology, you get a real sense of what's going on in the body and why that person is tired, why their adrenals are always weak, why they can't lose weight, why their histamine levels are higher than normal and always have been. Instead of giving antihistamines, get their body breaking it down, why they've been depressed, why they've been anxious. It's not a personal defect, and that was a big take home as well, as understanding that these genetic variants are not your fault. You inherited something here. We can work with it and change things, but one of the things that I did was I sort of beat myself up when I couldn't pull myself out of a depressive state, or when I couldn't get my energy going no matter what I did. I sort of beat myself up like it was a personal defect. Turns out, when I started upregulating my vitamin D receptor, my body started to make T cells correctly and fight infections. And any kind of SIBO, it'll clear up SIBO as well. So we don't have to go in there and work on small intestinal bowel overgrowth. The body will do that. You get rid of all the bad bugs out of your gut flora, and you've changed the microbiome. You don't have to go in there and micromanage the microbiome or leaky gut. The body will do it, and it's stunning when you do it this way. It was amazing. It blew me away when I started to see what was happening. So I upregulated my vitamin D receptor by taking vitamin K and vitamin A with my vitamin D, and then bam, my body started making dopamine, serotonin. My depression was gone, and I kid you not, within a week it was gone, and I felt happy for the first time in my life. I'd heard the word, and I thought there were times when I was happy in my life, and maybe there were. But to have a sense of like, that's who I am, that's where I live now, is a very different place than to just be working on my attitude and working on my mood all the time. And dopamine turned into norepinephrine and adrenaline, and my energy went through the roof, and I didn't have to work on my adrenals anymore. chronic fatigue syndrome left because now my body can turn on T cells and fight infections. And it changed everything. It changed everything for me. And I thought, there's something here. It got my attention. You know, when it works for you, it's like, oh, wow, there's something going on here. Because I had tried everything for 40 years, working on my health, and nothing worked. And I've tried a lot of stuff, and I know you guys have too. But it was a real eye-opener to look at the health this way. So I hope you'll join us on, let me see, I'm going to take you to where our website is here. Okay, so if you go to Wholistic Methylation, here's our contact info. Wholisticmethylation.com. You can buy the test kits there. You can look at our webinars and videos. We have all our downloadable forms there. To purchase the interactive chart, there's a link right there if you want to go directly to that. And, you know, gotmethylation.com, that's our tech support. Gotmethylation@gmail.com. General info at holisticmethylation@gmail.com. And you can join us on the Holistic Methylation Practitioners private site. So thank you again. I know that was a lot to kind of take in. We are putting all of our courses. There's a level 1, a level 2, and an advanced level 3 and certification available. So there's lots of learning here. We are putting it all online, so stay tuned there and go to holisticmethylation.com to see where we are in that process. My Happy Genes is where you go to get your DNA test done. And again, our DNA kit is very close to being ready for purchase. And we do have a practitioner's side to this program as well, where the software will actually calculate all this info for you and spit out a supplement program that you can use. So it makes life very easy, where you don't have to memorize all this biochemical data or genetic data. So lots of ways that you can use this information. So thank you once again, Dr. Belli, and everybody at the ICAK for being so generous and asking me to come and lecture. I will be answering as soon as I get this video uploaded again onto the site. I think they're going to send me the questions. So if you've watched this video and you have some questions, be assured that I will be answering those questions as soon as I can. Thank you so much for your attention. Bye-bye.